Hi everybody, I'm Jake Elmer. I'm an assistant professor from Villanova University, which is just a short, beautiful drive up I-95. It's about 2 hours from here in Philadelphia. But what I'm here today to talk to you about is a little project we've been working on lately, which is investigating the relationship between a host cell's transcriptome and its overall transfection efficiency. So that's a mouthful, but essentially what we're trying to get at here is we're trying to determine if there's any genetic basis for what determines if a cell is going to be relatively easy or difficult to transfect. After all, that's something that I'm sure a lot of us in this room have encountered before. You can take a given gene and put it into something like a HEC293 line, and a high percentage of the cells are transfected and they express a lot of protein. But then you take that same gene, put it into something like a T cell, and you'll find that you get a very low transfection efficiency, and even the cells that are transfected, they only make trace amounts of your protein. So what we're trying to do here is we're trying to leverage next-generation sequencing to analyze these different cell lines and see if we can pick the needles out of the haystack there to see if there's any genes that are, let's say, missing in HEC cells but present in T cells that would make it problematic. So just to clarify, first of all, we are a non-viral gene delivery lab, so everything I'm going to share with you today involves lipofection with lipofectamine. We have investigated PEI as well, but we're also going to be looking at ex vivo. So we are going to look at cancer cell lines exclusively here. So here are the 4 cell lines that we're going to include in this study. First of all, HEC293, wonderful positive control. We can get nearly 100% transfection of those cells. And then we're also going to include the endothelial lines PC3 and MCF7, prostate and breast cancer respectively. With those, we get about 30 to 40% transfection. And then our difficult to transfect line is going to be the Jurkat T-cell lymphoma. Now there with the Jurkats, we get about 22% of those cells that fluoresce after transfection with GFP. And even that has required a lot of optimization in our hands. For example, in that figure there, we're using 5 times the amount of lipofectamine with the Jurkats than we are with the other cell lines just to get to that level of expression. And even when we get that level of 22% of the cells fluorescing, notice there in the table on the bottom, we get about 1,000-fold less level of fluorescence. So not only are those T cells hard to transfect, but they're producing very little protein. So we've got a nice range here with these cell lines in both transfection efficiency and transgene expression. So it seems like a good way to pick out some differences there. Now, initially, a very important question to address would be, well, are you actually looking at differences in host cell gene expression here, or is it a delivery issue? And granted, a lot of the problems you see on this slide could be with delivery. I could be using a better vehicle here. For example, with the Jurkats, I can crank that up to about 80 to 90% transfection efficiency if I just electroporate them. But with lipofection, what we can show is that, oh, I really wish I had my pointer, but the images there on the right side show that if we co-transfect the Jurkats with 2 plasmids, one for GFP expression and another fluorescently labeled plasmid, what we see is only a few of those cells fluoresce with the GFP. But a lot of them show that they've taken up the fluorescent plasmid. So I'd like to argue here that yes, I might not be delivering to 100% of the cells, but there are certainly some of these cells that are receiving the plasmid, but they are not expressing the transgene. So if that's the case, what could be the underlying problem there? Well, something that's fairly well known already is that a lot of cells have an innate immune response to double-stranded DNA. After all, double-stranded DNA should only be in the nucleus, so any double-stranded DNA in the endosome, or let's say the cytosol, could be recognized as a sign of infection. It simply shouldn't be there. So for that reason, cells have these DNA sensor proteins, for example, very well-known TLR9 in the endosome that can recognize unmethylated CpG motifs there. And DHX9 and DHX36 pretty much do the same thing in the cytosol. They bind to DNA, then bind to MyD88 adapter protein, activate a signaling cascade of kinases there that culminates in the activation of transcription factors. Namely, AP1 and NFKB are the most commonly studied, but you can also see from this pathway activation of some interferon regulatory factors as well, especially ERF5 and ERF7, which then give you cytokine expression, ranging from TNFs to chemokines, interleukins, and interferons. But thanks to this evolutionary arms race that we've had between cells and viruses over the eons, the story doesn't stop here. The picture is much more complex, right? We can also get cytokine expression from these other DNA sensors. So cGAS, DBX41, DAI, they all exist in the cytoplasm. They can bind to double-stranded DNA and then activate the STING axis here. So STING, being localized to the ER, can activate TBK1, and you'll get expression of all these transcription factors along with ERF3. And then, oops, one too many animations. I'd like to point out that not only are these DNA sensors found in the cytosol, you can also find them in the nucleus, which is a complicated picture of how that works, of how they differentiate between host and foreign DNA. But sensors like IFI16, IFIX, and MNDA can recognize in those other compartments as well, double-stranded DNA there. We can also have LRRFIP, which is somewhat of an abstract DNA sensor, but it directly activates beta-catenin, which can then complex with TCF4 and cooperate with ERF3 to really drive expression of some specific cytokines, including interferon beta. We can also have some of these DNA sensors, including IFI16 and AIM2, that go on to activate the inflammasome. So they work along with caspases to induce things like pyroptosis, apoptosis, and secretion of some inflammatory cytokines like IL-1 and 18. So there we can have an immediate response to viral infection where the cell gets infected and the cell apoptoses to prevent viral replication. And then last but not least, there are actually some DNA sensors that I could not fit onto this slide. But the last one I'd like to mention is RNA polymerase III. As our last speaker mentioned, can actually bind to DNA in the cytoplasm, make some 5' triphosphate transcripts that then activate RIG-I, and then that goes right back into the STING TBK1 axis. So, a lot going on in this slide, but what I want to point out here is that we have a plethora of different DNA sensors that then activate a lot of different cytokines that can trigger this inflammatory response. And that response is characterized as such. These cytokines can go over to other cells that haven't even been transfected yet, for example, and we get a propagation of the response. So we can signal through MyD88 again, or some JAK-STATs to get even more cytokines. There's a variety of ways we can do that. Or if we focus in on the interferons, we can get expression of some interferon-stimulated genes as well. And now this is where things get really interesting, because now we start shifting away from just general inflammation and going into things that can actually affect viral replication. So for example, down in the lower right there, we can have oligoadenylate synthetases and ZAPs, which can directly degrade mRNA, or ISG15, protein kinase R, and the IFITs that can inhibit translation or target proteins for degradation. So this very redundant, resilient response is a great way for our cells to naturally defend themselves against viruses. Absolutely essential, but it's also a big stumbling block for gene therapy. Because what could happen here is I could have the best gene delivery vehicle in the world. I transfect 100% of my cells. But if I've activated all these pathways here, then my transgene might end up in the nucleus, and I end up with no protein expression, because one or more of these pathways is working against me. So that's the hypothesis here. But what we know from the literature, a quick survey, is that not all of these pathways are active in all cells. Some cells have, let's say, TLR9, some do not. So what we're going to do in this study is determine which of these pathways are important for each of these different cell lines. So we start off with JERCATs, MCF7s, and PC3s, and HEX, the ones I saw showed the FACS data for previously. We're going to transfect them without serum, just to eliminate any confounding effects there from the undefined media. And we're going to extract RNA at about 24 hours to survey for gene expression. Seemed like a pretty good time point there. Should be plenty of time for the cells to be expressing these interferon-stimulated genes. We're going to take that total RNA and fractionate it, do some poly-A selection to look at the protein coding genes, and some size separation to look at the microRNAs as well, because those could definitely play a pivotal role here. More on those later. Let's start by just looking at the mRNA sequencing data here. What I'd like to do is direct your attention first to the lower left there, the PC3 panel. What you see on the x-axis would be the gene expression levels in the control cells that have not been transfected. Y-axis, cells that have been transfected. So everything in the gray area there would be genes that have not changed at all. Anything orange or blue are genes that have been upregulated or turned on. Now, in the PC3s, we see a very potent innate immune response here. There are hundreds of genes that have been activated there. In the MCF7s, the breast cancer cell line, we see a lot of the same genes that are getting turned on, but the magnitude of upregulation is much smaller. So that's interesting. But then when we look at the HEC293s there in the upper left, we see essentially no upregulation of genes after transfection. A lot of the same genes that have been upregulated in either PC3s or MCF7s simply do not get activated in HEX. There are a few dots there, but they're unessential proteins, things that really don't have an antiviral function. So that's pretty exciting. Just from this 5,000-foot view here, we can see that there's a big phenotypic difference in these HEX cells that might be driving this higher transfection efficiency that we see with that cell line. But then we look to the lower right and the JERCATs. Now, the JERCATs were the cell line that were very difficult to transfect. We were only getting about 22% there. And the somewhat confusing thing there is that we don't see any upregulation of genes there either. So that was somewhat confusing initially, but then when we started to dig into that data a little bit deeper, we realized a lot of those orange dots you see there with the PC3, those genes that are upregulated in PC3s, are constitutively expressed in the JERCATs. So these JERCATs are already poised in an antiviral state, which makes a lot of sense, right? They're an immune cell, antiviral T cells. So they are ready whenever you transfect them. They don't have to go through this cytokine cascade where they turn on the genes. A lot of the effectors are already present, waiting to attack your transgene or prevent expression. Just a quick slide here. We're using lipofectamine. There is a chance that some of these genes that we're seeing could be due to the lipid itself. There have been some studies that have shown cationic lipids can bind to things like TLR2 and activate similar signal cascades. But what we did here is we repeated the transfection of the PC3 cells with PEI to see if we would get the same response, and surely enough we do. A lot of the genes that are upregulated in lipofectamine are also upregulated in the PEI model, and a lot of them to the same extent. They fit pretty nicely along that diagonal line there. However, it is worth mentioning that in this specific line, this PC3, TLR2 is a little bit low on the expression side, but even so, we do see higher expression of IL-6 with lipofectamine-mediated delivery than we do with PEI. That could be due to a lot of factors. Lipofectamine is a lot more efficient of a vehicle than PEI. We get about 2 times more delivery with lipo than PEI, but there you go. We're seeing a lot of the same genes. It's just a question of magnitude. If anything, the cationic lipid might be augmenting the response, but it's definitely not creating it. Okay, so now let's break those pathways down bit by bit by bit. Everybody took good notes and pictures of that previous slide, right? Okay, good. So let's start with the DNA sensors that center around the inflammasome. So for example, AIM2 and IFI16. If we look at that pathway specifically in all 4 cell lines, what we see is, oh, and by the way, I need to break this plot down here for you really quickly. What you're going to see is 4 different colored bars. JERCATs are red, and then MCF7's purple, PC3 blue, and HEX green. So if you see a color-coded bar there, that means that the gene was expressing in the control cells that were not transfected. And then any increase in expression is represented by a yellow bar on top of that. Okay? So if you see a blue bar with a yellow bar, that means that gene expressed in PC3, but it was significantly upregulated after transfection. Okay? With that in mind, what we notice with this inflammasome axis is that we really don't see those components in a lot of the cell lines, which isn't too surprising. I mean, when we transfect cells, they usually don't pop immediately, right? However, in the PC3 line, we do see quite a few of the components. So we see, for example, both AIM2 and IFI16, both of which can feed in there to ASC. We see the caspases and the interleukins as well. So there's a chance here that we might actually be killing some of the PC3s just by transfecting them. And surely enough, in that specific cell line, PC3, what you see on the right bars there is that after transfection, we see a significant increase in apoptosis with an annexin 5 propydium iodide state. This is in contrast to Jurkat cells. Now, all cationic vehicles are a little bit toxic, right? In the Jurkat cells, we see a lot of necrosis. That would be the increase in the red bar there. But in PC3s, we do see an increase in necrosis, but we see more of an increase in apoptosis. It's not the complete story. We're not killing all of the cells, but we are killing some of them. So this is something that's partially important there, at least with the PC3s, but it's not the whole story, so let's keep going. So next, we'll go to that LRFIP, the one that activates beta-catenin. Now what we see here is that a lot of the components of this pathway are present in all 4 cell lines. So you see that red, purple, blue, green kind of patterning for most of these genes, which isn't too surprising. Beta-catenin is involved in a lot of different pathways. ERF3 is constitutively expressed in most cells. It just has to be phosphorylated to be active. But there's a potential here that then in all of these cell lines, we could be seeing an LRFIP response. So what we did is we tried to chase that down with a small molecule inhibitor, ICRT14, that inhibits the interaction between beta-catenin and TCF4. TCF4 being a shorter peptide that allows beta-catenin to bind sequence-specific sites in the genome. So if beta-catenin can't bind TCF4, it can't do its job. And what we see is in PC3, Jurkat, and MCF7s, we see a significant increase in transgene expression when we add this inhibitor, probably because we're breaking up this pathway. So that was pretty neat. But we wanted to get creative with this, and we figured, okay, this highly suggests that beta-catenin and TCF4 are activating some transcription. Maybe we can utilize that. So we looked up the motif for TCF4, put that onto an EF1 alpha promoter, and what we find here is that at least in the PC3 cells, where we see, if I could highlight there, a significant increase in IFN beta, so that's what beta-catenin is doing. It's driving expression of the IFN beta in those cells. In those PC3 cells, if we add this TCF4 motif, we can increase transgene expression about 10-fold. This being luciferase expression, by the way. Unfortunately, we can do the same thing with GFP. It doesn't really help transfection efficiency, but in a transfected cell, we can get more transgene using that motif. Okay, here's where things get really complicated. Lots of bars. Sorry, I hope you brought your magnifying glasses and telescopes, but I'll try to summarize this as best I can. Oh, without my pointer. All right, so next, let's look at all those DNA sensors that funnel through MIDI88. So this would be the canonical DLR9, DHX9, and DHX36. What we find here, first of all, that's pretty interesting, on the left there, is that we don't see TLR9 expression in any of these cell types. We do see a slight increase in TLR9 after transfection, but it's so small you can't even see it on the plot there. However, we do see DHX9 and DHX36, so we could still be seeing some CPG sensing here. However, it's interesting to point out that you also see that in the HEX cells. These HEX cells that have a high transfection efficiency. So what's going on there? Well, if you take the next step down the pathway, DHX9 and 36 have to signal through MIDI88, which is present in the Jurkats, MCF7s, and PC3s, but completely absent in HEX. So that might be the crux of the model here, is that HEX are missing MIDI88. They might have all these other DNA sensors, but they're missing MIDI88, so they can't activate the pathway. In addition, HEX also appear to be missing IKKE, sorry. So they might have some weakened NFKB signaling as well. So that's pretty interesting. Maybe that lack of an innate immune response that we're seeing from the FPKM data and the higher transfection efficiency might be due to the absence of this MIDI88. But that's not the only signaling pathway, right? We also had that STING axis as well. But again, if we look at the HEX cells, and sorry for not having the laser pointer, but STING is over there above that orange rectangle. STING is missing in the HEC293 cells. They're lacking that adapter as well. And several of the DNA sensors shown there on the left. And another thing that's fascinating is those MCF7 cells. Remember, they had the same type of gene expression pattern as the PC3s, but it was at a much lower magnitude. MCF7s are also missing STING. They might have the MIDI88 axis, but the STING axis is non-functional for them. So that might be why we see that less potent response there with the MCF7s. But in PC3s, everything is alive and well. And that's probably why we see this potent response. You can see a lot of yellow bars there. Those are all PC3 responses right there. So not only do they have the necessary DNA sensors and adapters, but they also upregulate them after transfection. And one thing I'd like to point out about Jurkats is you see a lot of red bars there without yellow bars on top of them. A lot of these things are already present in Jurkats, ready, poised to defend themselves against viral infection. Oh, there's my notes right there. Okay, so these pathways are active. What kind of genes do they express? Well, let's start by looking at cyto and chemokines. First of all, this is a 24-hour time point analysis, so we don't see interferon alpha. That expresses a little bit earlier. But what we do see here is potent upregulation of IFN beta, IFN lambda, some interleukins and chemokines, all in PC3s. We see a little bit of this in MCF7, but this is most pronounced in PC3s. They are really churning out the cytokines here. Increases of 100,000-fold of these genes. And then we go on down to the interferon-stimulated genes. Remember, these are the effectors here that can actually limit viral replication. And what we find here is that, well, there's hundreds of these genes. It's truly marvelous what these cells can do. And it's a little overwhelming at first, but what you realize is that with these interferon-stimulated genes, you're activating the entire repertoire of the cell. They don't really worry about, well, am I facing a double-stranded DNA virus or an RNA virus? It just activates interferon-stimulated genes that defend against every type of virus. So a lot of these might not necessarily be important for double-stranded DNA. We're actually still in the process of knocking these out individually. Come back for my talk next year. That's what that'll be about. But what I'd like to point out here, what I've tried to do for today, is point out there's a few dozen genes here that are very interesting because they're upregulated in PC3 and MCF7, and they're constitutively expressed in Jurkats. So these are the most likely effectors here that really define those cell states, those low transfection efficiencies that we see in the Jurkats. These are antiviral proteins that are always expressed in the Jurkats. All right. So what can we do about this? Well, in the short term, one thing that we're trying to do is use small molecule inhibitors to chase down some of these pathways. Specifically in PC3 cells, we've definitely shown that the STING axis is very active. So we've tried to go through and identify small molecule inhibitors for a lot of different parts of this pathway, from the DNA sensors all the way down to the JAK-STAT signaling. And there's curcumin. And what we find here is that not all of these inhibitors work, and this makes a lot of sense because this pathway is so redundant. If you go after, let's say, a specific JAK-STAT signaling pathway here, there might be another one to compensate for it. So AG490, nifuroxazide really don't work. But if we hit on a bottleneck, so something like inhibiting STING itself, that most of the DNA sensors have to go through with H151, that small molecule inhibitor, we can see a significant increase in transgene expression, about 3-fold there. And if you recall from the GFP FACS experiments, the table showed that there was about a 4-fold decrease in GFP expression from HEC to PC3s. Here we're seeing a 3 to 4-fold increase in that expression. Okay, so that's what we've seen from the protein coding genes. Now I'd like to switch gears a little bit here in the 2 minutes I have left to explain microRNA biology. But the basic idea here is that while you have some genes that code for protein, you can have other genes that code for microRNAs, and they work in the opposite direction. They actually repress protein expression. So typically, you get a microRNA that binds to something like AGO2 there. It'll patrol the cytoplasm looking for any transcripts that are complementary, imperfectly or perfectly, and if it binds to something, it'll repress it. That's the basic idea there. So I could have microRNAs that are being activated here that either drive the inflammatory state by repressing anti-inflammatory proteins, or what I'm really afraid of is that I could have some microRNAs that are directly targeting my transcript and repressing transgene expression. So we've done the small RNA sequencing in all the lines, but this is all I have time to show you here. The PC3s are the most interesting. What we see here is that we get at least 4 microRNAs that are significantly upregulated following transfection. Out of those 4 that are in that little green oval there, One of them is miR-155, and this is something that's been known for a while. miR-155 is involved in the innate immune response. So what it will do is it will repress expression of anti-inflammatory proteins like SOX1. So we'll get more potent JAK-STAT signaling if we have miR-155 expressing there. So miR-155, we see upregulation of that in the PC3s. And guess what? We see constitutive expression of that in the JurCats. So again, JurCats are always poised, ready to defend themselves against virus. But then one last thing that I'll leave you with is that one of those blue dots there in the green oval is a microRNA that specifically targets the 3' UTR, the plasmid that we were using. Now, the 3' UTR we're using is the rabbit globin poly A, so it's not too surprising that there would be a microRNA for this. It's a globin, so it's going to be repressed at some times. But we see a good seed region matching there, so this is a bona fide target or a match there. But what I find really fascinating is that not only does this microRNA exist, but it is upregulated after transfection. So the big question here is this an active type of response to the transgene itself? Okay, so I'm out of time, so I won't summarize, but HEC293s, easy to transfect because they lack an innate immune response. PC3s have a big fat one, MCF7s much less, JurCats, all of their effector proteins are always expressed, and that's how they resist viruses. All right, with that, I'd like to thank my lab group, especially Matt Tucker there on the right and Emily Harris in the middle. They collected a lot of the mRNA sequencing data that I showed today. And Matt Hall, a very outstanding undergrad, did some cloning work and small molecule inhibitor work that I showed today as well. Thanks to my collaborators and NSF, and thank you all for coming and standing up in the back of the room. Okay, so I think there were two parts to the question. One was payload size, or burden, or how much transgene we're delivering. Was that the question?
The specific transgene that you are actually delivering, does that have an impact?
Oh.
Instead of reporter.
So we've only looked at GFP in these expressions. Or no, actually we have looked at both GFP and luciferase. That was some of the PEI data that I showed earlier, and the response is identical. I think there was a question in there about the amount of gene that we're delivering. Okay. So there's a lot of things that could happen there. If we're talking about AAV or another virus, we could be circumventing some of these DNA sensors. We could escape some of them or avoid them and go directly into the nucleus. And that could definitely change the landscape there, because then you're potentially not activating STING, but the scary thing is that still IFI-16 could recognize your plasmid in the nucleus as well.
Nice presentation.
Thank you.
Two questions. The first thing is you showed different cancer types, but if you take, there are, even within the same organ, if you say PC3, there are different versions of the prostate, and they're hard to transfect or expression levels vary. So have you compared within the same organ type, different cell types, to see if there are differences? The second one is the JurCat T cells. Are they IL-2 activated, or they're just without activation that you're seeing the differences?
So, easy question first. JurCats have not been IL-2 activated. With the different cell types inside the same tissue, that's something we have planned next. Because now that we have the next generation sequencing done, we have the targets. Now we can do a wider screen RT-squared PCR to see if these genes are expressing in different cell types.
Thank you.
Please.
Are you using the conventional 293, right? Without the large T antigen, right?
Sorry, what's that?
293s or 293Ts with a large T antigen?
293T.
That's relevant because that protein is what degrades in situ. Also, have you tested some primary cells? Have similar results?
Primaries are hard. What's limiting us right now with the primaries is I really want to look at primary T cells. But if I want to continue this avenue of research, lipofectamine is only giving us around 4 to 5% transfection of those primary T cells. So we need to be higher than that to really get a good glimpse of the transcriptome in those cells. We have it planned. We're trying some things. If nothing else, we're going to do it with electroporation later this summer. But we're not there just yet.
Jacob, I have a
Oh, yes.
quick question. Do you think the lipofectamine is eventually, is going to be relevant to in vivo gene transfer down the line? Or would it be, I mean, do you think the responses to lipofectamine will mirror what responses you would have towards viruses, let's say, or a nanoparticle, even in your system?
So hopefully what we're looking at here is just delivery of the plasmid DNA. And a lot of these results would carry over no matter what vehicle you're using. Maybe, maybe not. But I'm primarily interested in ex vivo gene therapy where you could still use lipofectamine. But like I said, we're still trying to push the envelope there to get it to a point where it's competitive to electroporation and retroviral transduction. Not there yet.
Hi.
Yes.
As you know, HEK293s were transformed with adenovirus, and adenovirus E1A, E1B are known to impact a lot of these upstream, detection, including the STING pathway. Have you thought about looking at the effect of those genes on the other cell lines to see how much those genes by themselves might actually dampen the innate response?
Right, there's a lot of ways we can go about this, right? One would be co-expressing different genes, and that's something you could easily package into any viral or any vector you're thinking about using. You could put in E1A or something like that to subvert parts of this response. That's what the viruses do. That's what's so beautiful about this. That's why the innate immune response is so complex, is because viruses have figured out how to inhibit it with these different proteins.
And as a follow-up question, have you looked at the downstream genes, the ApoBEC3 genes that activate the cytosine deaminases that actually destroy the plasmid and the incoming DNA? Have you looked at those guys are being turned on in your cells in response to the
We don't see
transfection?
much of a response there. We have seen some interesting things with DNases, though. A lot of those do get upregulated.
Okay, great. Thanks, everybody. I'll have to ask the questions to go on afterwards because there's a line forming outside the door for the next session. Thanks for the speakers and thanks for everybody showing up.